For thousands of years, romantic attraction has been framed as a mystical force or a poetic condition. But when observed through the lens of clinical physiology, the onset of attraction triggers a measurable biological shift. This is an aggressive, highly predictable survival drive that mirrors the clinical states of mania or chemical dependency. The precise neural mechanisms behind this drive remained largely theoretical until 2005. That year, anthropologists and neuroscientists used functional magnetic resonance imaging to scan the brains of young adults who reported being intensely, passionately in love. When these subjects viewed photographs of their romantic partners, the scans didn't highlight cortical areas associated with complex social reasoning. Instead, a massive surge of blood flow targeted the right ventral tegmental area, a tiny structure nestled deep within the brain's primitive reptilian core. The ventral tegmental area acts as a manufacturing hub for dopamine. While popularly called the feel-good chemical, clinical neuroscience defines dopamine as the primary engine for motivation, craving, and reward prediction. When a powerful external drug like cocaine is introduced to the body, it forces this exact neural network to dump massive amounts of dopamine, creating an overwhelming craving. The brain scans of the impassioned lovers displayed this identical pattern, the same primitive pathway lighting up in anticipation of a reward. This specialized chemical routing dictates what the organism perceives as vital. The brain executes a highly coordinated motivational program, categorizing the romantic partner as an absolute biological necessity. While dopamine explains the high physical energy and relentless drive to pursue a partner, it doesn't account for the darker realities of new love. The literal inability to stop thinking about a person, often at the expense of sleep or basic obligations, requires a disruption in an entirely different system. In 1999, researchers analyzed the blood chemistry of people in the early stages of infatuation. They found a severe depletion in the density of serotonin transporters. The levels matched perfectly with unmedicated psychiatric patients diagnosed with severe obsessive-compulsive disorder. Without serotonin to act as a cognitive brake, dopamine-driven anticipation spirals into continuous, unchecked fixation. This diagram illustrates the neurological architecture of early-stage attraction. Notice the massive focal deactivation in the prefrontal cortex, responsible for logical reasoning and critical judgment. Simultaneously, we see deactivation of the amygdala, the primary alarm system for processing fear and threat detection. The phrase, "love is blind," describes a measurable, biologically enforced suppression. By actively silencing the areas that regulate social caution and logical analysis, evolutionary biology ensures that two self-preserving humans bypass their natural interpersonal defenses long enough to successfully mate. To understand why the brain utilizes such extreme neurochemical measures, we have to view human attachment through the selective pressures of our ancestral environment on the African savanna. Early hominins faced a severe biomechanical conflict. As they evolved obligate bipedalism to efficiently navigate the changing ecology, the pelvic canal necessarily narrowed to support upright walking mechanics. Simultaneously, unique evolutionary pressures selected for rapid encephalization, resulting in progressively larger, highly complex brains. This created the obstetric dilemma. Females could no longer give birth to neurologically mature offspring. Human infants had to be born profoundly helpless and premature compared to virtually all other mammals. A human infant requires years of constant, calorie-heavy care just to survive predators and the elements. The metabolic cost of raising a large-brained child far exceeded the foraging capacity of a single female. Romantic obsession evolved as a highly specialized attraction system to solve this biological bottleneck. It compelled a male and a female to hyper-focus their mating energy exclusively on one another, conserving immense metabolic resources and ensuring dual parental protection during the offspring's most vulnerable years. This frantic biological imperative relies on a high dopamine, high cortisol state. But that level of physiological stress and extreme cognitive distraction is ultimately unsustainable for long-term health and survival. To prevent systemic burnout, biology designed a secondary system. The chaotic energy of initial attraction is eventually mediated by two ancient neuropeptides, oxytocin and vasopressin. Released in significant quantities during physical intimacy, these molecules interact directly with the deepest pleasure centers. This coordinated activity permanently links the sensory signature of a specific partner to a lifelong source of homeostatic comfort. As relationships mature into secure attachment, the brain's cognitive brakes are fully restored. Cortisol normalizes, the obsessive serotonin deficit resolves, and the fear-processing amygdala reactivates, trading the anxiety of early infatuation for profound physiological security. Decades of marriage are sustained by persistent activation in the dopamine-rich ventral pallidum. Long-term devotion operates on this permanent neuroplastic change within the forebrain's reward circuitry, driving cooperation and mutual well-being long after the initial obsession fades. Because social connection was a non-negotiable requirement for hominin survival, the human brain developed a brutally efficient alarm system. Extensive neuroimaging studies prove that the brain processes romantic rejection and social isolation using the exact same neural pathways that register literal physical tissue damage. When a bond is suddenly severed, the extreme emotional stress can cause the autonomic nervous system to panic. This triggers a toxic catecholamine storm, dumping a massive, uncoordinated surge of adrenaline and norepinephrine directly into the bloodstream. This flood of stress hormones binds excessively to receptors concentrated in the heart muscle. The apex of the heart's left ventricle can become temporarily paralyzed, violently struggling to pump blood as it balloons outward, generating clinical symptoms identical to a massive myocardial infarction. This medical diagram charts the progression of Takotsubo cardiomyopathy, a severe and sometimes fatal condition where profound psychological grief induces acute myocardial toxicity and left ventricular dysfunction. The condition takes its name from the distinct abnormal shape the deformed heart assumes, which closely mirrors a traditional Japanese pot called a takotsubo, utilized by fishermen to trap octopuses. Heartbreak manifests as a profound systemic shock. The body utilizes the agonizing sensation of pain to alert the individual to the life-threatening danger of social isolation, compelling them to repair the severed bond, just as they would tend to a bleeding wound. Modern humans are currently deploying this sensitive, ancient circuitry in a radically unnatural environment. The digital landscape of algorithmic matchmaking effectively gamifies the human mating drive. Dating applications exploit a foundational mechanism known as the seeking system, an ancient neural network that rewards mammals for exploring their environment. By deliberately staggering matches and messages, the underlying algorithm places the user on a variable ratio reinforcement schedule. This is the exact intermittent reinforcement design pioneered by behaviorist B.F. Skinner. The deliberate unpredictability of rewards causes massive, erratic spikes in dopamine, creating a compulsive behavioral loop where the endless anticipation of a mate substitutes for genuine human connection. Digital behaviors uniquely facilitated by this anonymity, such as sudden, unexplained withdrawals of communication, wreak absolute havoc on the attachment system. This triggers violent cortisol spikes due to the evolutionary threat of social rejection, completely denying the user the stabilizing oxytocin resolution of a secure physical pair bond. The neurochemical architecture of human bonding dictates the parameters of our deepest attachments. It systematically dismantles logical defenses and hijacks core reward circuitry to ensure independent individuals bond long enough to rear altricial offspring. It remains one of the most powerful, highly conserved survival systems in nature.